All right, good morning, everyone. Welcome to our second round of awardee presentations. I'm looking forward to hearing some of the exceptional work that we're going to hear this morning. Again, my thanks to the Beacon staff and the entire study section for helping put together the best work and to bring it for you today. So this morning we're going to start with Dr. Weisi Liu, who is a postdoctoral fellow in hematology and oncology at Weill Medical College of Cornell University. Dr. Liu is a senior postdoctoral fellow in the Faltis Lab at Weill Cornell Medicine. He is focusing his efforts on studying the mechanisms and downstream effects of ABOBEC-induced mutagenesis in urothelial carcinoma and developing therapeutic strategies to target the mutagenic fuel of bladder cancer cells. Dr. Liu has published in peer-reviewed journals and recently received a training grant from Anvil Cloud Credits Continue Program, or the AC3, from the National Human Genome Research Institute. And his title for today's talk is Synthetic Lethal Strategies to Limit Tumor Evolution Driven by ABOBEC Activity in Urothelial Cancer. Dr. Liu.
Thank you, Peter's introduction. Good morning, everyone. So my name is Weisi Liu. Upgrade a little bit. I'm right now the instructor in the Dr. Faltis Lab at Cornell Medicine. So it's a great honor to stand here to present our current work named Targeting MMEJ by inhibiting polypsita in the urothelial cancer with ABOBEC3 activity. So brief introduction ABOBEC3 ABOBEC enzymes. So these enzymes have 11 members in humans, and these enzymes stimulate the cytidine in the preferred TC motif in the single-stranded DNA and RNA. So they will result in the C2T or C2G mutation in the DNA due to the DNA repair mechanism or CTU editing in the RNA. So these enzymes, the main functional is immunorelated. So they were involved in the virus restriction and the diversity of our antibodies. And also, they are related to the metabolism using their RNA editing function. By detecting the mutation footprints in our human cancer genomes, ABOBEC3-induced mutations are highly enriched in the multiple cancer types, including the bladder cancer. That's indicated that the cancer cells hijacked these enzymes as a mutagenic fuel to increase the tumor evolutions. Our peers' paper have been found that the ABOBEC3-induced mutations are highly enriched in the chemotherapy-resistant cancer patient samples. And recently, the paper found that the therapy itself can trigger the ABOBEC3A expression and drives the evolution on these survival cells. That would be said because that means every time you give the patients with the treatment, and meanwhile you will increase the opportunity to get a resistance to this treatment because ABOBEC induced the tumor evolutions. So what can we do? Is there a small molecular inhibitor can targeting the ABOBEC3 enzymes? The answer is no. Unfortunately, there is no currently inhibitions in the market can specifically target ABOBEC3 enzyme. So is there an alternative way to target the downstream, the DNA repair mechanism to restrict the ABOBEC3 activity and also illuminate the cancer cells? So to address these clinical questions, we need to answer first, does ABOBEC3 induce the lethal DNA damage in our cancer cells? Second, what's the preferential downstream, the DNA repair mechanism for the ABOBEC-induced the lethal damage? Number three, is inhibition this repair mechanism is synthetically lethal in the cancer cells with ABOBEC3 activity? So to answer this first question, we generation a cell line model using in the 5637 cells using the doxycycline-induced ABOBEC3A. So we choose ABOBEC3A because the previous paper found that ABOBEC3A is the major contributor to the ABOBEC mutation in the bladder cancer. So we turn on the ABOBEC3A for the 24 and the 48 hours. And then we check the replication stress marker and the double-stranded DNA break markers. As we're showing here, you can see the tumor cells when the exposure to the ABOBEC3A, the CHK1 and RPA is highly activation in the cells. That's indicated that the cells have the replication stress. And also the marker of the ATM, the CHK2 and the gamma H2X, also highly activated in the cells. That's indicated the double-stranded DNA break. So that's data indicated that the APOBAX3A can cause the replication stress and the lethal DNA damage, double-stranded DNA break. So how the APOBAX3A induce the replication stress since they just deaminated cytidine as a single-based mutation? So to answer this question, we're using the DNA fiber assay to identify the impact of the APOBAX3A activity on the replication fork. So this DNA fiber assay using two different thymidine analogs. So these analogs can integration into the DNA when the replication process. And then we can using the antibody to specific labeling this DNA fragment. When we compare the ratio between these two fragments, as we're showing here in the right, so you can judgment the replication status is ongoing or is terminated. So we did this experimental in our cell line model, as we're showing here with the represented image and the quantification data. It's clearly that the APOBAX3A decreased the ratio of these two fragments, indicated that the replication fork is terminated, and then might be the reason triggering the replication stress and accumulation of the double-stranded DNA breaks. So right now we know APOBAX can cause the lethal damage. So what's the preferential downstream, the DNA repair mechanism for these damages? Before I answer these questions, we need to know that there are the three major double-stranded DNA break repair mechanisms in our cells. One is called the classical NHEJ, another two is HR repair and MMEJ repair. So since the APOBAX-induced double-stranded DNA breaks happens in the replication phase, so we focus on the HR and the MMEJ repair because these two mechanisms is predominant in this phase response to the double-stranded DNA breaks. As I highlighted here, so the single-stranded DNA overhangs at the break's end, binding by the RPA is the essential steps to make the decision choice for the repair. So when the single-stranded DNA overhangs, binding by the RPA, and then the RPA replaced by the RD51, so the repair will go to the HR repair using the system chromosome as a template. But if they have the limited RPA displacement, so they will exposure the DNA to overhands and then promote annealing using the microhomologous sequencing, and then the repair will go to the MMEJ mediated by the polyceta. Of course, the MMEJ repair is the error-prone repair process, so that's why we'll create the unique mutation footprint called the microhomologous mediated deletions in our genomes. So is that APOBAX3A can significantly increase the microhomologous mediated deletions. So we're sequencing our cells, the cells model. So these cells already exposure to the APOBAX3A over 2 months. And then as you can see here in the left, the APOBAX3A expression cells have significantly higher number in the indels, indicated that the error-prone DNA repair mechanism is recruited. And also after the classification definition by the cosmic, we find the microhomologous mediated deletions also significant in our cells. So to validation our experimental finding in the human samples, we did a whole genome sequencing in our patient cohort. So as is showing here in the left, there a significant correlation between the APOBAX-induced mutation burden and the microhomologous mediated deletions. And we also found that this significant correlation is not only happen in the bladder cancer, it also happens in another cancer types when we're using the TCGA pan-cancer whole exome data as we're showing in the left. So this result indicated that these two mutation footprints consistently occurs in the human cancers. So we know, right now we know that MMEJ is preferred to respond to the double-stranded DNA break caused by the APOBAX3A. But why is MMEJ? Why not is the HR repair? Does the APOBAX3A will direct involve into this decision maker? So as I mentioned before, the RPA binding to the single-stranded DNA overhand is essential steps for the choice, repair choice. And also the APOBAX3A can binding the single-stranded DNA overhands. So we have some hypothesis that APOBAX3A can displace the RPA, resulting in the expression of microhomologous sequencing and promote MMEJ repair mediated by the polyceta. So to answer these questions, we designed a biochemical assay as we're showing here. We're using 2 oligos to mimic the break ends. So the longer one on the 5 ends will label with the fluorescence marker for our detector. And also the 3 ends will have the microhomologous sequencing, and in the middle is the motif for the apobac binding. And then we mix these oligos with the A3A, RPA, and the polycystic proteins. So in the reaction, if the apobac 3A overcompete with RPA, so they will exposure more microhomologous sequencing, and then they will annealing, and then the MMEJ polycystic will involved in to generation the longer annealing products. But if the RPA is overcompete with apobac 3A, so the microhomologous sequencing will be protect. Sorry about that. Where we go? Okay. So when the RPA overcompetes with the apobac 3A, so RPA will binding the single-strand DNA over hands and protect the microhomologous sequencing. So they will inhibit the MMEJ repair mediated by the polycystic. So when we do the reaction and run the gel, so here is the gel image. So the upper band is the annealing products, and the bottom band is the unannealing products. As we highlight here on the left, you can see when we added the polycystic into the reaction, so they were generating more the annealing products as we expect. But when we added the RPA into the reaction, it's highlighted in the middle, so the products will decrease because the RPA protect the microhomologous sequencing at the 2 ends. And when we added the apobac 3A into the reaction, the annealing products reversed, significantly increased. So here is the quantification outcome. That data indicated that the competition between the apobac 3A and the RPA for the single-stranded DNA overhands on the 2 ends, resulting the annealing or the microhomologous sequencing and promote to the preferential to the MMEJ repair. So right now we know the MMEJ repair is the downstream double-stranded DNA break repair mechanism to the lethal damage caused by the apobac 3A. So is there the inhibition, the polycystic, the key protein in the MMEJ is synthetic lethal in the tumor cells with apobac 3 activity? To answer this question, we go back to our cell line model. So we knock down the polycystic using the shRNA. As we're showing here in the left, it's the knockdown efficiency using the qPCR by the 2 different primer sets. And then we turn on the A3A. As we're showing here in the right, with the apobac 3A increase their expression level, the polycystic knockdown cells are much more sensitivity to the apobac 3A induced toxicity compared to the SH control cells. So we want to test is that this effect also can be used in the clinical. So we recruit 2 different polycystic inhibitors to identify the synergistic effect between the apobac 3A expression level and the polycystic inhibitors. So we recruit one drug called novobiocin. So it's targeting the ATP binding domain. Another drug is called ART558. It's targeting the polymerase domain. So these 2 drugs right now is in the clinical trial. So we performed the cross-experimental with the dose curves of these 2, with the doxycycline, which indicated the apobac 3A expression level. It's on the x-axis, and also the concentration dose curves with the drug, the polycystic inhibitors. Here is the synergy map. So the red area indicated the concentration of these 2 effects are synergistic, and the green one indicated the opposite direction. So we're using the ZIP model to calculation the synergy score, and these 2 scores indicated that there is a synergistic effect between the apobac 3A expression level and the polycystic inhibitions. So we're still working on to the experimental validation our synthetic lethality in our organoid model and the patient-derived xenografts model. I think the missing one slide, but it's fine. So in conclusion, so basically we found that the APOBAC3A can induce the lethal double-stranded DNA breaks in the cancer cells. And the second, we found that the polyceta-mediated MMEJ is the predominant downstream DNA repair mechanism response to this lethal damage. And also we found that targeting the polyceta is synthetic lethal in the cancer cells with APOBAC3 activity. So at the end, thank you for my mentor, Dr. Feltus, their support, and also all the members in the Feltus lab, and also thank all the collaborations for their efforts. And of course, thank you to the support from the Beacon. So I'm happy to take any questions. Thank you.
So I had a quick question regarding, and I don't know the DNA repair pathways perhaps as well as I should, but we know that in bladder cancer, and we've heard about a variety of different germline mutations, BRCA1, BRCA2, ATM, et cetera, et cetera, et cetera. Do you have any sense yet of how those types of mutations could interact with this? Because, in a lot of these cells, there's going to be more than one aberration. So how do those two interact? And how do you think that's going to affect your results or impact how you use this ultimately clinically?
That's very great questions. So currently, these two inhibitors for the MMEJ repair is tested in the breast cancer with the BRCA mutation. And also, we know they have patients in the bladder cancer also have the BRCA mutation. So I'm thinking if the patients have the BRCA mutation and APOBAC's high burden, high activity, which will be more sensitivity to these drugs. So we're still using this filter to try to figure out in our organoids. Mm-hmm. So we classify them with the organoids like APOBAC mutation, if they have the mutation in the BRCA or not, or another DNA repair mechanism to test their response to the drugs. So we're still working on this.
Okay, because I would anticipate that the mutational profile within the different organoids that you're testing might very well influence their response to these.
Yeah, exactly. Yes.
And actually that offers a potential opportunity to actually have predictive biomarkers developed as part of this process, which is actually really exciting. And it could be a first, really, in many respects.
Yes, exactly.
That's awesome.
Thank you.
Go ahead, Bill.
Oh, sorry. You're looking over there.
Just making sure there wasn't somebody else, that's all.
So I think Charles Swanton's group has published that, at least I believe primarily in cell lines, there's more episodic expression of APOBEC, and so I'm curious what your thoughts are about in vivo human tumors. Is the episodic expression reflected in that? And if it's truly episodic, then does that prohibit your synthetic lethal approach if there's not actual APOBEC3 activity ongoing in a tumor?
I just rephrase your question. So you're thinking because our model is using the overexpression system, so you're thinking in the patients, the expression level will not be that higher enough to create more damage. Is that correct?
So there was a paper that suggested that the expression of APOBEC, albeit in cell lines, is episodic, meaning it's not always constantly mutagenizing, but it's this sort of intermittent activity that's driving the mutagenic process, which is perhaps reasonable given the replication stress you're showing and the inability of cells to actually tolerate that long term. So what I'm curious about is your thoughts, if this is true. So I think we actually, if anybody wants to take it on, I think we should actually look at human tumors, bladder tumors over time and see if there is truly APOBEC activity that's differentially regulated over time. But I don't think we actually have, maybe your replication stress assay is nice, but I was thinking the classic deaminase assay isn't really that sensitive, at least in our hands. And so okay. I'm curious whether you think this is a problem for your Pol theta inhibition strategy.
Yeah, that's a great question. So first we test in our organoids with the APOBEC mutation burden to predict is there reaction, the response to this polyceta inhibitions. And then meanwhile, as I show in the paper, the therapy can also induce the APOBEC A3 expression level. So this drug is the EGFR inhibitors. And in their paper, they showed clearly that when you give the cells with these inhibitors, they will cause the double-stranded DNA break using the H2X markers. And then when we knock down the ApoBac3A, the double-stranded DNA markers decreased. So that means, so if we also will, that's good chance if we do not see any good response when we just test in the organoids, we might be a combination with certain treatment. So that will maybe after this treatment, they trigger in this expression level, and then we can follow up with the polycystin inhibitor. So we can use in the combination with these two drugs in a clinical trial to see the patient's reaction, and also you can enlarge the response with the drugs, also limited the evolution to the residents.
Yeah, and the other only thought is, it's clearly like a very nicely upregulated by interferons. So, maybe the steroidin adds, whatever you call it, the add interferon, that kind of thing may also be a nice adjuvant. So
Yeah.
Very nice work, though, nonetheless. Thank you.
I echo Billy, great nice work. Congratulations to you and the team and Bishoy. I was actually looking at, recently there was a paper in lung cancer looking at ApoBec signature, the mutagenesis signature, correlating with prognosis and potential response to immunotherapy. Any comments about, based on the mechanism we nicely showed, prognostic role and potential predictive value, just following up Peter's question, in response to platinum-based chemo or immunotherapy, or other therapies in bladder cancer? Again, great work.
I think we're still working on this. So it's good to using this marker to detect the reaction with the chemotherapy as a biomarker. But considering the ApoBacs will cause tons of the evolutions, so it might be harder to using all the burden to reaction the chemo, or specific target, say this will cause the resistance or not, because ApoBac create a lot of mutation. Some may be just a passage mutation, some may be as a driver. So we still need to working on to identification which one is the driver to the chemo resistance, and then that will maybe improve our predictor marker to predict the reaction to the therapy. It may be harder just using the ApoBacs or the mutation burden. That's my thinking.
Thank you.
Thank you.
Until we have an effective inhibitor, are there certain agents that are less likely to induce more ApoBac activity? For instance, taxanes, which are microtubular agents, versus alkylating agents, which cause double-strand breaks. Is there a hierarchy of known chemotherapeutics or other drugs that are more likely to induce this accelerated activity?
That's a great question. I think it could be, because the chemotherapy also will create a lot of the DNA damage, but also the issue is when you create a lot of the damage by the chemo, they also will create opportunity for the ApoBacs, for the single-stranded DNA break, right? So that may be also the chance we can combination with the chemo with this polycystin inhibitors.
I was wondering, since your group has published on the induction of ApoBac activity by chemotherapeutics, whether or not there's a hierarchy of drugs more likely to induce it than others. A listing of known chemotherapeutics and their ability to upregulate ApoBac. Oh, very good. Thank you.
Yeah. Thank you.
Great talk. I'll ask for Bishoy. I'll ask if it's true, what does it imply question. So if there's typically thousands or maybe hundreds of thousands of ApoBec lesions in a typical bladder cancer. So does that mean each site of ApoBec alterations was a double-stranded break, like prior to its activity, prior to the ApoBec? I mean, that means that there's like many, like, I guess, did we realize that there's that many double-stranded breaks prior to a ApoBec mediated mutagenesis, or are there potentially other ways that ApoBec may have activity in the absence of a double-stranded break?
I think I'm not fully clear with the question. So you want to ask, is that another mechanism?
I guess the real question is, does ApoBec3 activity, at sites of ApoBec3 activity, are they always preceded by a double-stranded break, or can ApoBec work in the absence of a double-stranded break?
That's a good question. So, as B.D. King said, the APOBACs need high expression level. That will cause double-stranded DNA break. So they need to accumulation certain of the mutation burdens that will stop the replication fork. So we have another mechanism can bypass this AP sites caused by the APOBAC. So they will cause the mutation, single-base mutation, but still can go through the replication force, which may not cause double-stranded DNA break. So that's why this therapy or the treatment need more APOBAC activity to create a double-stranded DNA break. But in another side, so if you give the chemo or another DNA break reagents, they will create more double-stranded DNA break. In our experimental, we're thinking if that's the case, so you get more break ends, the APOBACs also will be involved in this decision choice. So that maybe also will be the sensitivity to the polytheta inhibitors.
So you're saying agents that cause double-stranded breaks may induce APOBAC3 activity then?
I think so.
So radiation and other, DNA damaging agents would you might hypothesize would have a similar APOBAC3 inducing activity then?
Yes, so that's with paper our mentor published in the Nature Genetics said the chemotherapy resistance the patients have highly enriched APOBAC mutation burdens.
Okay, thank you.
Thank you.